Hi, everyone. My name is Ashlee Brady, and the title of my project is Surface Water Quality of the Sandy Lick Creek in Jefferson and Clearfield County. And my advisor was Dr. McElroy. So these images are from this summer of me out collecting my water samples. This one is me collecting the water. Then there's one of me in my kayak going to get a sample. And then this last picture is just a picture of the stream that I thought was nice. So a little bit about my survey area. The Sandy Lick Creek runs through the town of DuBois and surrounding areas. And this, you can kind of see it starts way up here and then goes the whole way through and it ends down there, but it keeps going. But the Sandy Lick is highly affected by acid mine drainage, or AMD, and other anthropogenic activities, because as you can see, like, it does run through the town of DuBois, and out here, there's a water treatment plant and it runs right through the middle of town. So there's lots of storefronts and obviously roads and different things will run off and affect the water quality. My focus on the study was more up around where the DuBois Mall is, because in my last slide, I had an image, and that is actually an image of an old mine shaft that used to sit where the mall is now. So the water is incredibly orange up around the mall, and all these streams and the Sandy Lick that runs through there is very orange and bad with acid mine drainage. And so those were some of my first top points where I was mainly focused at around the mall. And then I wanted points basically after it ran through the mall and different things like that, close to the town of Falls Creek. And I just wanted to see if it got better or worse through town and just different stuff like that. So here's some images that were taken this summer. This image in the top corner here, this was my sample number 6, which I will talk more about a little bit later. But as you can see, it's very orange, and it was gross, and it looks almost like a thick sludge. I actually was not able to get a full bottle of water from this sample because it was just thick sludge, essentially. But so that was my number 6. And this middle image here, you can see, is me taking readings from a water sample from a stream up around by the mall. This middle image that runs through all of these here, as you can see, it's very orange. It's very gross still. It's highly affected with iron. And this last image is just the GPS that I use to get my latitude and longitude coordinates. And that can be used for if somebody else were to come out and take measurements like I have. So we use 4 analytical chemistry methods to test the water with. And the first one is suppressed anion chromatography, otherwise known as IC. And this provides quantitative measurement of various analytes or salts in the water. And these include chlorides, nitrates, sulfates, phosphates, and others. And this is an image of what the IC looks like. We use gravimetric total dissolved solids, or TDS, and this provides information about the overall amount of all dissolved substances in the water, which includes salts and other substances like debris and stuff like that. But this first image is a large hot plate. So we take 100 milliliters of filtered sample and we put it in these ceramic weigh bowls, which are then set on this large hot plate at 250 degrees Celsius. And once almost all of the water is evaporated out, except there's enough left to cover the very bottom of the bowl, they are then transferred into this oven to finish the evaporation process. And then they will be cooled and weighed. This next test is alkalinity titration, which basically just provides information about the stream's alkalinity and how much acid load the water can handle. This is the basic setup for the alkalinity, and in here would be hydrochloric acid, and it goes to the range of about 4.5, and that's the cutoff range for alkalinity. And the last test we use is inductively coupled plasma optical emission spectrometry, or ICP-OES. This machine provides quantitative measurements of 12 different metal species in the water. It calculates both major metals such as iron or calcium and minor metals like arsenic or cadmium. And this is an image of what it looks like. Here are some of my results. So I put all my results in this nice table, and I labeled it with sample ID, the date of which the sample was taken, my TDS results, alkalinity, IC results, and then each analyte, and then the ICP results with all the 12 metals. But as you can see, there are, for the IC results, it's like a 0 at 5, and then the analyte is named. Essentially, that analyte, and as you can see, it was a very high in chloride. So essentially, that sample needed to be diluted 5 times or 10 times, as you can see down here. And because it was too high and it was above our limit of 100 parts per million. But 6, as you can see, was diluted heavily from the very beginning, because if you remember from my image, it was the one that was sludge, it was gross, and we were not willing to take the risk of running 6 through the machines to we would risk possibly damaging the internal mechanics of the machines. So we diluted 6 from the very beginning just to avoid any possible things like that. And you can also see there are some different, like if the analyte or metal was detected, they were reported as is, but there's also some other things you may see on here, such as DNQ, which stands for detectable but not quantifiable, which means there was enough of the analyte fluoride in the water that it was detected, but we cannot assign it a quantifiable number, because it's right in the middle. So it may be there, but we just can't assign it a number or a value. Then you will also see less than LD, which is it's less than limit detected. So what this means is it's essentially not there. So we cannot reliably say that that analyte or metal is detectable in the water, because the machine may also pick up on noise or just some little vibration, and that might have given it a signal, but it's essentially, we cannot say that it is in the water for certain or not. And then here's just some more of my results. And I have 13 as a special talking point, because when I went out into the field to sample 12 and 13, I didn't realize at the time that when I crossed the little stream of 12 to get to 13 or further down the stream, that I didn't go far enough down the other side of the bank. I had essentially sampled 12 twice. So as you can see on May 23rd, that these results for 13 and then the very first 12 are very, very similar, meaning that I had essentially sampled 12 twice, but I didn't realize that until I was back in the lab and had discussed this and talked about the different scenarios and things that had happened. So then when I went back out, I labeled it 13A because it was no longer in the same place. And I took my kayak and floated further down on the main drag of the Sandy Lick. And then I was able to get better readings because I had let the stream or 12 mix in right there. So I was getting both of them then at 13A. So there's that. But for some main conclusions, the stream was a lot better than I thought it was. It is still heavily affected by acid mine drainage, as you can see from the orange colors in the pictures. And yes, there was reportable iron amounts in the stream, and it did have other things in it as well. Also, I want to note that due to time crunches, only some of the ICP results are included in the presentation. My last sampling trip was not included due to just time conflicts and stuff like that. But all of the data will be put in a report and given to Dr. McElroy once it is analyzed and ran. But hopefully in the future, another student may take up my project and pick up where I left off. I do have all the GPS coordinates written down in a notebook that they could use, and it should be very easy. But also, hopefully, like depending on some of this, like the results, that I may be able to take it to somebody in the town and say, hey, like I think we should focus on cleaning this up. So that'll be really cool, and hopefully I'll be able to do that. But my last thing is, I want to say a very big thank you and shout out to Alex Clark for taking the time out of his schedule to train me on the proper lab etiquette and techniques needed for this summer's research project, because I haven't taken analytical chemistry yet and just different stuff like that. So I did need trained on a lot of this stuff, and thankfully he was able to do that for me. So I would like to thank him and give him a shout out. But that's all for my presentation. Thank you for listening, and have a fantastic rest of your day.